Okay, hello everyone. We just have a quick couple things to go over before your test this week. This is something I'm pretty sure I lectured on, but I want to make sure that I didn't miss it in the previous lecture I posted. The test cross, because this is going to be a test question. A Punnett square, what we will do is we will do a test cross, which is where we take an individual that is a homozygous recessive and we cross it with someone that has the dominant phenotype. And I do remember we talked about this and I compared it to me and my husband, because if we assumed, which this is actually not accurate, that blue eyes are recessive to brown eyes, and that was all the eye colors that we could choose from, then that we would be an example of a test cross, because I have brown eyes and you don't know if hidden underneath those brown eyes is a recessive allele. And so what you do is you would cross with a homozygous recessive with the dominant phenotype, and what you would see is if any of the offspring end up showing up with the recessive phenotype, then you know that there was a recessive allele hiding underneath, okay? All right, so I'm going to skip forward through this question. This is just showing you the genetics of chickens, but we're not going to really cover that. So we're just going to kind of skip through. Now, here's the kind of exception I was talking about. Are genes always dominant and recessive? Is it always the simplistic fact that one gene is completely dominant and masks the effects of the other gene? No. Actually, genetics is much more complicated than that. And a lot of times genes are neither dominant nor recessive. And you have two different things. Well, the two different examples that we're going to go over in this class is incomplete dominance or codominance. Now, what you see here is that this is an example of incomplete dominance, meaning that neither gene gets to dominate over the other, and it is a shared effect. So what you see happen is that when you combine and here we don't use little, you can use a little r, but when we combine the two alleles, you get the intermediate, which would be a pink, okay? So you have a red and you have a white gene, and if it was normal Mendelian genetics, the red and the white, if they were together, one would be dominant over the other. So you would see a red, let's say red was dominant, a red flower with a little r hiding underneath. But with something like incomplete dominance, it's a blend of the two phenotypes, okay? So here we are with incomplete dominance. The heterozygote form has a phenotype that is in between the two homozygous forms, okay? So with incomplete, it means that they have a blending effect, okay? Neither allele gets to be completely dominant over the other, all right? Now we have another example called codominance. Codominance means that both alleles are expressed equally strong and they're visible. So it isn't, the heterozygote is not really an in-between. It would be where you would see a chicken, and if you have the black and the white genes, you end up with black and white coloring on the chicken. It wouldn't be a gray chicken. That would be an example of incomplete dominance. Instead, you would have both alleles expressed, but they would be expressed. That meaning you'd have black and white, or this is very random, but you could have a black and white polka dotted chicken, okay? Now, both codominance and incomplete dominance are an example where we aren't following Mendel's rules. And on your test, you will be held accountable for one of these. And what I would really encourage you to do is go through your test review and make sure that you define all of those concepts and terms, and that will be your homework for this day, would be to go through your test review and make sure that you understand all the terms and email me or ask me questions on any terms that you don't understand. So again, codominance would be where both alleles are expressed. You actually see them. So the chicken example would be if you have the black and the white genetics, you'd get a black and white speckled or a black with white dots or something, okay? If this was an example of incomplete dominance, then you would end up with a gray chicken. Now, here we have the cows. This is another great example. You have the white cow and you have the red cow. Codominance means that the red allele gets expressed and the white allele gets expressed. So you see both expressed here, okay? If this were to be incomplete dominance, that's kind of where you get the word. Codominance means they're sharing, right? They're sharing expression. Incomplete means that they blend. So these two, the white and the red, would make a pink cow, which we haven't really seen yet. So this is the dihybrid Punnett square. This is going to be in your notes. You're not going to be held accountable for this on your test. This is a cross that will track two characteristics. So what are the chances in the pea plants of the pea plants being green and wrinkled, or yellow and smooth, or yellow and wrinkled? The tricky part to this, it follows the same rules as the normal Punnett square, but the tricky part is setting up the gametes. And again, this is not going to show up on your test. I am going to include a video that talks about setting up the dihybrid Punnett square. Those of you that are going to take AP Bio might want to review that video so that you have an idea of how to set up a dihybrid cross, but you're not going to be tested on it, and I haven't even asked you any questions on your worksheets over it. Pedigree symbols, we're going to go ahead and skip because we're not going to be doing that for this unit. So, all of this, we won't be held accountable. And I just kind of wanted to make sure that you knew that for your test. So again, go through your test review, go through those terms, make sure that you can find all the answers in your notes. You need to be very competent at setting up a monohybrid cross and that would be this kind of a cross where you have four squares, right? And the mom and the dad are on the sides, and then the offspring are in the middle. You need to feel really good about setting up. That's what we call a monohybrid, where we're tracking one trait. All right, guys, good luck and have a great week.